Hello everyone, our today's topic for discussion is transcription. So, transcription is a process in which RNA is synthesized from DNA, and there are three different stages of transcription. The first is initiation, the second is elongation, and the third is termination. So, the first step is the initiation, and the initiation occurs when the RNA polymerase binds to DNA at the promoter region. This is the promoter region. And RNA polymerase is of various types, like RNA polymerase first, which is used for the synthesis of ribosomal RNA, the RNA polymerase second, which is used for the synthesis of the messenger RNA, and the RNA polymerase third, which is used for the synthesis of tRNA. So, for the synthesis of mRNA, we can use the RNA polymerase second, and this RNA polymerase second binds to this promoter site. In this, the promoter site is present at the 5' end of the DNA, and this is the termination site, which is present at the 3' end of the DNA. And in the center there is the structural gene or the transcription unit. Now, when the RNA polymerase second binds at this promoter site, it opens the DNA and forms the transcriptional bubble. And in the transcriptional bubble, only one strand is used for the transcription, and this strand is known as the template strand. And this strand always runs from the 3' to the 5' direction, because the RNA polymerase can only synthesize the new strand in the 5' to 3' direction. Now, when the RNA polymerase bind to DNA at the promoter region, and it is important that the 6 nucleotides base, that is TAT is the origin site of the transcription, and this is also known as the Tata box or the Pribnow box. So, the RNA polymerase is bind at this promoter site when it reads the Tata box or the Pribnow box in the promoter site. Now, when the RNA polymerase binds to the promoter region, a factor which is known as the sigma factor. This is the RNA polymerase, and this is the sigma factor, which is released, which is a part of the RNA polymerase. Now, the RNA is synthesized from the 5' to the 3' direction. The 5' to the 3' direction anti-parallel to the DNA. This is the DNA, this is the transcriptional bubble, this is the promoter region, this is the termination region, and this is the structural gene. So, here the formation of the RNA will take place from the 5' to 3' direction after the release of the sigma factor. This sigma factor is a part of the RNA polymerase. This is the RNA polymerase. So, here there is the elongation of the RNA takes place, and it is important that no primer is required for the activity of the RNA polymerase. Also, the RNA polymerase cannot proofread the mistakes of the RNA synthesis. So, these are the various steps of the elongation, in which various nucleotide bases will add to the RNA from the 5' to the 3' direction, and after the full elongation of the RNA, there is a process of termination at the termination site. The termination signals are given to stop the synthesis of RNA from the DNA. A protein which is known as the Rho factor binds to the RNA at the termination site and terminate the transcription and releases this RNA. This is the newly synthesized RNA. The Rho factor is also responsible for the dissociation of the RNA polymerase. The Rho factor is also responsible for the dissociation of the RNA polymerase from the DNA. So, these are the different steps of the transcription, starting from the binding of the RNA polymerase to the promoter site. Then there are the steps of the elongation, and then there is a step of the termination by the release of the Rho factor, and the initiation will only takes place when the sigma factor is released through the RNA polymerase. So, these are the different steps of the transcription. After transcription, there is the post-transcriptional modification. The RNA which is produced during transcription are called as a primary transcripts, and they undergo many changes such as the addition of the methylene group, base modification, splicing, etc. And these post-translational modification convert the RNA into the active form. The ribonucleases are the enzymes which are responsible for the processing of RNA. So, what are the changes which can occur in RNA, like 5' capping, poly A tail, introns and its remove. So, the changes which may occur is like 5' capping. At the 5' end of the mRNA is added by the 7-methyl guanosine, which is required for the translation and stabilizing the mRNA. Then there is poly A tail. After the synthesis of RNA, a polyadenine tail is added at 3' end, and this is also required for stabilizing the mRNA. Introns and its removal. There are many nucleotide sequences which do not code for the proteins, and these are called as introns, and they are removed by the small ribonucleoprotein particles. So, this is an outline of the post-translational modification and post-transcriptional. This is an outline for the post-transcriptional modification, and the post-transcriptional modification is important for the examination point of view. So, this is the capping, this is the 5' capping, and this is the poly A tail. Both of these were added to the mRNA for its stability. At the 5' end of the RNA is added by the 7-methyl guanosine, which is required for the translation and the stability of mRNA. This poly A tail is a polyadenine tail is added at the 3' end, and it is also required for the stabilizing the mRNA. The introns are removed. The introns are the nucleotide sequences which do not code for the protein. So, introns are removed by the splicing process. By the cutting process, various pieces which are unwanted can be cut, and there is also chemical modifications, like new chemical groups were added to this mRNA. Now, this mRNA which is obtained is functional unit of the mRNA. So, this is all about transcription. If you like this video, please do subscribe my channel and share with your friends. Thank you for your time.